Good morning, students. We will start with the active site of the enzyme. So the active site will be present in the enzyme. This is the site where the substrate binds and gets converted into product. This active site is also called as catalytic site. And this active site has three different points of interaction or contact will be made between the substrate and the enzyme's active site. And this was stated by Oxton in 1948. The three different points of interaction will always exist between enzyme and substrate so as to favor the enzyme substrate interaction. For favoring the substrate enzyme interaction, the ES interaction will require two types of complementarity. One is geometric complementarity, the other one is electronic complementarity. So these both complementarities are very important for the enzyme and the substrate to be in the biological conformation and to have the interaction between each other. So in the substrate, we have the reacting group and also the binding group. Whereas in the enzyme, you have binding group as well as the catalytic group. So binding group of your substrate and the binding group of your enzyme has to interact so as to have a proper binding orientation, which is stated by means of geometric complementarity. And the reacting group, which is present in the substrate, has to interact with the catalytic group, which is existing in the enzyme. So these 3 points of interaction will make the effective enzyme substrate complex. And this effective enzyme substrate complex can go ahead with the formation of product and the enzyme. So these three interactions are very important for the further theories, which also stated the mechanism of enzyme catalysis. There are two major theories for interaction between the active site of the enzyme as well as the substrate. The first hypothesis is lock and key hypothesis. This lock and key hypothesis, which is stated by Fisher in around 1890, and he stated that the enzyme and the substrate structure will be exactly complementary to each other so that the substrate can fit effectively into the enzyme. So the structure of the active site of your enzyme and the substrate will be exactly complementary. So which makes sure the geometric complementarity so as to make your enzyme and the substrate complex to interact with each other so as to obtain the product and the free enzyme. So later on, this kind of hypothesis cannot fit with all the enzymes because certain enzyme structure is different for its specific substrate. So in order to prove that, the next theory was proposed by means of Koshland in 1958, which is called as induced fit hypothesis. Enzymes in free solution will not be complementarity as such to the substrate, but when a substrate approaches the enzyme, the conformation of the enzyme will change in such a way that it just fits into the substrate molecule. So here the substrate is rigid and the substrate structure cannot be altered and cannot be changed. Whereas the enzyme structure is floppy in nature, the enzyme just goes near the substrate and it just wrap around the substrate, and according to the substrate structure, the enzyme will alter or change its conformation so as to make the enzyme to be very complementary to the substrate. So once if such kind of interaction happens between the enzyme and the substrate, now the enzyme substrate complex will be formed so as to have the interaction, thereby the product and enzyme will be released. So the enzyme will have the original conformation, which is not complementary to the substrate after the release of the product. So this hypothesis could be able to fit most of the enzymatic reaction which is happening in the biological system. So both lock and key hypothesis as well as the induced fit hypothesis plays a major role in the interaction between the active site of the enzyme and the substrate.